Today we're going to look at gene linkage and abnormalities. Before the lecture begins, make sure that you've read the assigned portion of your textbook for this lecture and taken notes on that reading. Make sure that you've prepared for yourself an outline to take notes during the lecture by looking ahead at the PDF. And finally, make sure that you're listening in a quiet space that's free of distractions, turning off any noise, excess music, and any extra devices. Today we're going to consider gene linkage and altered chromosomes. When we consider gene linkage, we need to remember that genes are located near each other on the same chromosome, tend to be inherited together in genetic crosses called linked genes. An example is in Drosophila, the fruit fly. Both wing size and body color tend to be inherited together, as is shown in the figure on the right. Genetic recombination is the production of offspring with a combination that's different than that of the parental gene. To see how linkage works, let's look at some specific genes. Two of the genes, 1 and 2, are relatively far apart in the top illustration. Each gene comes in two different versions or alleles, A and B. Since gene 1 and gene 2 are far apart, it is likely that a recombination event will happen between them. When this happens, the gametes end up with new allele combinations that were not present in the parent. That is 1B with 2A and 1A with 2B. Gene 3 and 4 in the middle illustration also come in two alleles each, A and B. But because these genes sit much closer together, it is less likely that a recombination event will happen between them. Remember that the location of chromosome breaks during recombination is random. So most of the time, 3A and 4A will stay together, and 3B and 4B will stay together. Therefore, genes 3 and 4 are linked. Genes on separate chromosomes, such as genes 5 and 6, are never linked, looking at the bottom illustration. Each gamete gets a single copy, determined at random, of each chromosome. Because there is nothing holding them together, these alleles can pass to gametes in any combination. Researchers can use linkage to find the location of a gene on a chromosome. By looking at how often different genes are inherited together, researchers can create maps of the relative distance between these genes. Since each gamete gets one of two possible versions of a chromosome by random chance, two unlinked genes will be inherited together 50% of the time. Unlinked genes may be on different chromosomes or so far apart on the same chromosome that they are often separated by recombination. If two genes are inherited together more than 50% of the time, there is evidence that they are linked on the same chromosome. The closer together the genes are, the more frequently they will be inherited together. Now that we've considered gene linkage, let's look at altered chromosomes. Large-scale chromosomal alterations in humans and in other mammals can often lead to spontaneous abortions known as miscarriages. Large-scale chromosomal alterations can also cause a variety of developmental disorders. Plants tolerate such genetic changes far better than animals do. In non-disjunction, pairs of homologous chromosomes do not separate normally during meiosis. This results in one of the large-scale chromosomal alterations that can impact development. During non-disjunction, as a result, one gamete receives two of the same type of chromosome, and another gamete receives no copy. In the figure on the slide, you can look at several non-disjunction events. In figure A, there's a non-disjunction of homologous chromosomes in meiosis 1, which results in daughter cells from the cell on the far left having odd copies of chromosomes. In the non-disjunction of sister chromatids in meiosis II, you can see that both cells end up with odd daughter cell numbers of chromosomes. Aneuploidy results from fertilization involving gametes in which non-disjunction occurred. Offspring with this condition have abnormal numbers of certain chromosomes. A monosomatic zygote has only one copy of particular chromosomes. A trisomatic zygote has 3 copies of a particular chromosome. And polyploidy is a condition in which organisms have more than 2 complete sets of chromosomes. Triploidy is known as 3 sets of chromosomes, and tetraploidy is known as 4 sets of chromosomes. So triploidy is a certain type of polyploidy, and it results in a 3n number of chromosomes. Tetraploidy, again, a certain set of polyploidy, which results in a 4n set of chromosomes. Again, polyploidy is common in plants. It's better tolerated there, but it's not common in animals. Polyploids are more normal in appearance than aneuploids. So now that we've looked at an altered number of chromosomes, let's look at alterations in gene structure. Breakages of chromosomes can lead to 4 types of changes in chromosome structure: deletion, duplication, inversion, and translocation. Deletion removes a chromosomal segment. Duplication repeats a chromosomal segment. Inversion reverses the orientation of a segment within a chromosome. And translocation moves the segment from one chromosome to another. Examples of each of these can be seen in the figure on the slide. Figure A shows the deletion, which is the removal of a chromosomal segment. Figure B shows the duplication, where there's a repeat of a segment. C shows an inversion, which reverses the segment within the chromosome. And D shows a translocation, a movement of the segment from one chromosome to a non-homologous chromosome. When we're looking at gene structure, a diploid embryo is homozygous for a large deletion is likely to be missing essential genes. Such a condition is generally lethal. Duplications and translocations also tend to be harmful. Inversions, the balance of gene is normal, but the phenotype might be influenced if the expression of the gene is altered. So next, let's consider what happens with the altered chromosomes. Alterations of chromosome number and structure are associated with some very serious disorders. Some types of aneuploidy upset the genetic balance less than others, and this can result in individuals surviving to birth and beyond. These surviving individuals have symptoms or a syndrome that's characteristic of the type of aneuploidy that's present. Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. Down syndrome affects about 1 out of 830 children born in the United States. The frequency of Down syndrome increases with the age of mother. But this correlation between the age of the mother and the frequency has not yet been explained. Non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is a result of an extra chromosome in a male producing an XXY individual. About 1 in every 1,000 males are born with an extra Y chromosome, and these individuals do not necessarily exhibit any defined syndrome. Females with trisomy X have no unusual physical features except for being slightly taller than average. Monosomy X is called Turner syndrome, and this produces XO females who are sterile. It is the only viable monosomy in humans. So today we've covered gene linkage and altered chromosomes and what happens when altered chromosomes are altered in number or there are alterations in gene structure. If you have any additional questions, please feel free to reach out to your professor through email or visiting during office hours. Additionally, you can reach out to your learning assistant who's assigned to this lecture. Combine your notes from this lecture with the notes from your assigned reading. Use the assisted reading questions to test your knowledge of this material, as well as the questions at the end of the chapter and at the end of the chapter sections.